I'm going to take you through my AP Biology genetics packet. This specifically goes over Mendelian genetics. This first page is pretty easy. It's going over things like alleles and genotypes. It says number 1, circle the alleles below that are dominant and draw a square around the recessive alleles. So again, this first one, you just draw a circle around it because it has the capital letter and then a square around those recessive alleles. For number 2, you're showing the genotype for both the homozygous dominant, the heterozygous, and the homozygous recessive genotypes. Like for this first one, it'd be big Y, big Y, big Y, little y, and then little y, little y. You do that just for each letter. Over here, you're just identifying whether each genotype is heterozygous with an HE or homozygous with an HO. So this first one would be homozygous. This next one would be heterozygous. This one over here would also be homozygous, even though they're different, like capital versus recessive letters. They're still both going to be homozygous. Number 4 asks you to identify what genotypes will produce the dominant phenotype and what genotypes will produce the recessive phenotypes. So this first one produces the dominant phenotype, and the heterozygous forms will also produce the dominant phenotype. It's not until you have 2 recessive or lowercase letters that you're going to produce the recessive genotype. Number 5 asks you to, giving the genotypes, you have to produce what the phenotype would be. So like purple flowers are dominant to white flowers. Big P would be purple flowers. Big P, little p would be purple flowers. And then little p would be the recessive white flowers. And the opposite of that, we're going back from the phenotype to the alleles. It doesn't give you specific alleles to use. So you can just use whatever alleles. I just go with A and B just to start out. So big A, little a, and then little a, little a. Number 7 asks you how to, or what the chance of having a heterozygous, homozygous recessive, or homozygous dominant parent passing on the dominant recessive alleles. This is just getting you to think when the parent passes on a single allele, what are the chances of that occurring? This next part here, you can see each of the new pages that I kind of separate the packet into has a bolded title. These are monohybrid crosses, and these monohybrid crosses are just going to give you an example of what's going on in the question. What you have to do is you have to tell what the zygote possibilities or what this Punnett square can produce. So I usually put like this one, I think is A, so it's gene for big A is dominant over the dwarf little a in the garden pea plant used by Mendel. So like the big A, how many of these would be big A? How many of these would be big A, little a? How many of these would be little a? I would write the ratio, so like 1 out of 4 equals 25%. I do both the ratio and percent. Then tell me what the phenotypes are. So like in this one, like what percent of the offspring are going to be tall? What are the offspring are going to be dwarf? You do this for a couple problems. You can see again that goes through a couple of these problems. The next page here, this is page 4, I think. This is independent assortment. You can see here, this is asking you to identify what the genotypes or what the gametes will have. And for example, this first one, there's only one because you have to give a single letter from each of these genotypes, like big A, big A, big B, big B. You're going to give one of those A's and one of those B's, and the only combination is big A, big B. However, for like this one, you have 2 combinations because it could be big A, big B, little c, or little a, big B, little c. And this is just taking you through independent assortment and crossing over and saying, hey, what's occurring between these and what are the possible gametes? Now, this one, number 18, is a little bit more confusing. This is asking you to separate these chromosomes through meiosis 1 and then meiosis 2. And I always tell students, make sure you draw the chromosomes as well, because that's going to really help you out. Now, I print these out in like black and white. They're not in color, so my students don't see this color, but I show them on my sheet what it looks like. And it's really important for the students to know that this independent assortment, it allows us to create the differences in the gamete. So like in this case, we have a big R, big Y, little y. But the recombinant DNA would be, I should say really recombinant, but the other, I should say, chromosome possibility or genotype possibility would be big R, little y, and little r, big Y. So it shows you at this end, we have 4 that are actually in these 2 are going to be the same, these 2 are going to be the same, these 2 are going to be the same, and these 2 are going to be the same. That leads us into the dihybrid crosses here. This is just showing you a cross between 2 heterozygous parents, and it's asking you to go through that cross, and you can see dominant for yellow peas is big A, recessive for green peas is little b, dominant for smooth surface is big B, and then recessive allele for wrinkled surface is little b. And I ask you to do the dihybrid Punnett square. And I always tell my kids this is the only dihybrid Punnett square you should ever do in my class because of the next part here. And then it asks you what genotypes are possible to create yellow and smooth. I think there should be 4, if I'm remembering correctly. There should be 4 here. There should be 2 different possibilities for both of these. There's only 1 possibility at the end, and you'll understand why after you go through this Punnett square. Then it asks the ratio of the offspring of the yellow and smooth, so how many will there be? And this should be out of 16 for each of them, and then what that probability is, so what percent that is. Product and sum rules. This goes over how to get through a dihybrid Punnett square, or a trihybrid, or you can see a quad hybrid without actually producing one. And this is called the sum, or I'm losing my words here. Sum and product rule, I think goes over this. So after your teacher, or after I go through this, you'll understand how to go through this really easily. These are pretty easy once you get a hang of them. But again, you're just multiplying and then finding the percent of like a certain type of offspring. So like in this case, big A, little a, big, or little b, little c, big D, little d, out of these 2 combinations of the parents. All right, we're almost done here. So autosomal dominant versus recessive genetics. This just asks you to identify like whether it's an autosomal dominant or recessive type of inheritance, and then possibilities and genotypes of parents and offspring. This is pretty, you can work through this pretty easily. And lastly, genetic recombination problems. Man, these can be tough at the beginning. And I always tell my students, I really don't care if you can get through one of these problems. They should be able to get through a couple of these problems. Like this one's a little bit of it's difficult. But I always tell my students, like, you should be able to tell me which 2 are going to be inherited more often together and which one are going to be recombined more often. Because we're looking at the locus, or the gene location of specific genes. And again, this is a nice tool to have, but not really necessary for the AP test or like a CHS test. And that is, I think, the end. Yep, that's the end of this is the Mendelian genetics packet.